Our first speaker today is the one and only Dr. Paul Grossfeld. He's a board-certified pediatric cardiologist and a clinical professor of pediatrics at the University of California, San Diego. Dr. Grossfeld takes care of patients with congenital heart disease at Rady Children's Hospital in San Diego. He saw his first patient with Jacobsen Syndrome in 1995, and since that time, he's assembled a multidisciplinary group of investigators studying genetic basis of congenital heart defects and Jacobsen Syndrome. Dr. Grossfeld serves as the Chief Medical Advisor for the U.S., Spain, and European Jacobsen Syndrome group. He has reached out to colleagues both locally in San Diego and from around the world to bring together a team of clinical experts to help with the various aspects of care for individuals with Jacobsen Syndrome. He's co-authored over 20 peer-reviewed scientific papers on Jacobsen Syndrome. He lives in San Diego with his wife, Susan, and 17-year-old son, Stefan. In his spare time, he loves to travel internationally, play a variety of sports, and swim and snorkel in nearby La Jolla Cove in the summer. And that's his formal introduction, but we all get to call him Dr. Paul. He's amazing for our organization. We can call on him whenever we need to. I know many of you have taken the opportunity to email him, call on him, have him contact your doctors for advice. And on top of that, he's one of the primary planners of this conference. So we're so excited to have him speak to us today. Without further ado.
Thank you. All right, how's everybody doing? Yeah, good. Halfway through. Padres won again last night, so you guys are bringing them good luck. If they win again tonight, they sweep the series, and it's got to be because of your team, so that's great. All right. One quick announcement. Yesterday, I went around and I took a picture of every one of your boards, posters, and I thought, if you guys are okay with this, I will hire my son, my IT consultant, to take all the pictures of each person's poster into like a PowerPoint presentation that we could send out to everybody. And that way you could have it as like a little memento of the conference for all the families. If any of you, you don't have to tell me now, but if any of you are not comfortable allowing me to do that, to share it with the group, just let me know, let me know individually, but I think that'd be a really nice way, because I don't know about you, there's so much fascinating information and so many incredible stories. I always feel like I can't catch it all just during the conference, so this way we'll kind of make that a permanent memory. So before I get started, if anybody has any questions during the talk, please feel free to stop. It's informal. I'm going to go through some fairly technical stuff, so don't get worried about that, but hopefully you'll kind of appreciate the big picture. And I give a fair amount of talks at conferences locally and nationally, even internationally, but what's really special is when I look over here, this is my inspiration, the Servin family who lost their baby this past year to Jacobsen Syndrome here in San Diego. I'll try not to get emotional up here, but it's really touching. And then right next to them is Harlan, who was born with hypoplastic left heart syndrome. And it's like your stories are what inspires me along with the rest of you. But, unlike any other talk I give, this is such a unique interaction. So thank you guys for being here. And I know for the Servin family, with everything you've been through, it's just remarkable that you come here and be part of this group that you will always be part of. So, okay, let's see if I can remember how to make this go. So what I want to do is focus on a few of the heart defects that we've been studying in my lab. This is just kind of an overview of what I'm going to go over. So you've heard me talk about, this is okay, Harlan, for me to divulge your heart defect, hypoplastic left heart syndrome, and how we have identified some genes now that are on 11q that we think are critical in terms of their function in different types of heart cells that are required for heart development. So the idea is when you lose the function at the critical time in the critical place, meaning that in a specific population of cells, we're trying to learn how that causes the heart defects that occur in your children. And hopefully, ultimately, that's going to help us to improve care, not just for people with Jacobsen syndrome, but maybe even the more general population of people with defects like that occur in Jacobsen syndrome. And I'll talk about a new gene that we've recently discovered that one of the people in my lab, Marjan, you don't need to see me, you just need to see the screen, that's fine. Because I can't see it. And you'll see how that kind of relates to the story of the genes that we've already identified in 11q. And again, I just can't emphasize enough how grateful I am to all of you and the other organizations that have been so supportive over the years to keep my research going. The biggest challenge is I've had grants from other agencies like the NIH and American Heart Association, but those come and go. And the greatest challenge is to have sustained funding, and that's what you guys, many of you have been successful in doing. That's allowed me to keep things going. One other person that I failed to mention the other day is Rich Siritz, who is our treasurer, who has been so awesome and instrumental in helping to coordinate the logistics of when you guys make donations. So I don't know if Rich is watching today, but just for the record to acknowledge his incredible support and efforts. So some of the questions that I'm going to come back to that have kind of evolved over the years, in part from listening to you guys and just learning, is why is it that you could have Parlin with one deletion and another person with the same exact deletion, Parlin has hypoplastic left heart syndrome, and someone else with the identical deletion may have a completely normal heart. And I think we're beginning to understand why that might be, which is really interesting. I'll come back to that. And similar to that, whether or not you have a heart defect, and the next question is, why do some people have hypoplastic left heart syndrome and someone else with the same deletion may have something a little less complicated, like maybe just a simple hole in the heart? And I think we do have some answers to that. Another one that I'll talk about is total anomalous pulmonary venous return. Who's the family? I don't know if they're here, whose child was born with total anomalous pulmonary veins. They're one of two families. Elliot Thomas is the other one. And I'll show you how we think we can understand why one person might get hypoplastic left heart syndrome, another might get anomalous veins, and another may have a completely normal heart. And just kind of for fun, every so often people are curious about, why does it seem like so many of your kids have red hair? And I think you might have an answer to that, just for fun. Okay, so as you heard me talk the other day, as I learned more about the syndrome, there were probably 3 causes that stood out in terms of the most common or most frequent causes of death in children with Jacobsen syndrome. Heart problems, which I'll talk about today, which still remains a challenge. Bleeding issues and infection issues, and thanks again to Chuck Haymeyer and some of our other people that have participated now. As I hope all of you know, we have those medical protocols that you can hand to your treating physicians who are legally obligated to follow those. And with that awareness, I think we're making a significant impact on reducing the chances of anybody suffering from those. So really, my hope is nobody moving forward should ever die from a bleeding complication or from an infection with Jacobsen syndrome if we can get this information out there. So coming back to hypoplastic left heart syndrome, this is kind of the typical cartoon picture that you'll see of hypoplastic left heart syndrome. Carlin, feel free to correct me if I say anything wrong. Okay. So what you see here on the left is a normal heart. So this is the left ventricle. So that's the main pumping chamber that pumps blood out to the body through the largest vessel called the aorta, shown here in red. And you can also see here, this is a valve that's wide open. That's called the mitral valve, which allows blood to flow to the left ventricle to get pumped out to the body across the aortic valve. So in hypoplastic left heart syndrome, what you have is a much smaller left ventricular chamber or cavity, so that chamber is not able to adequately pump out enough blood to the body. And most of the time associated with that small chamber is a problem with the valves. In this case, the mitral valve and the aortic valve are too small. Sometimes they're so abnormal that they actually can't open at all. And that's called atresia, versus being small but still able to be open, that's called stenosis. And just to jump ahead a little bit, what we learned in Jacobsen syndrome, and I'll show you in detail in a moment, is in most cases of hypoplastic left heart syndrome, the mitral and aortic valve are completely blocked off, so they don't open at all. And if you look at comparing to the normal, you could imagine that the reason why this chamber is so diminished is because there's a thickening of this heart tissue that makes up the left ventricle. And we actually know, if you look at that under a microscope, that is because there are too many cells. And this raises the interesting issue that, which I think illustrates why there's been so much confusion in the field, because people call it hypoplastic left heart syndrome. And in this case, what you can imagine is there's growth or abnormal growth inward to kind of obliterate that. Does that make sense? But in the real world, we actually see different anatomic versions or subtypes. So the one I just described to you is, this is the left ventricle of a heart, hypoplastic left heart. So this is all that massively thickened wall that the cartoon just showed you. This is actually what was supposed to be that chamber. And in this case, you tend to see scarring lining that chamber. In Jacobsen syndrome, what we actually see is something you could say is completely opposite, shown here. So in this case, rather than having a massively thickened left ventricle, we actually see a very diminished left ventricle, what we sometimes refer to as a slit-like left ventricle. And in this case, in most of the cases, the two valves on that side are completely blocked off. So we call that mitral and aortic valve atresia with a slit-like left ventricle. And this is actually from one of the babies who passed away last year, showing with hypoplastic left heart syndrome and Jacobsen syndrome, this is what was supposed to be her left ventricular chamber, so totally slit-like. And so even though we treat this the same surgically, in reality, I think that the mechanisms are very different between these different anatomic subtypes. So when there's too little growth, we are pretty sure now that that is due to something that happens very early in heart development, and I'll come back to that in a moment, versus the other situation where there's overgrowth, that actually we think can happen much later in heart development. And I think intuitively, if we can understand what's going on at the molecular and cellular level, distinguishing these two mechanisms, we're going to be able to come up with better ways to manage these patients than how we do it now. Speaking of which, how do we do it now? Well, Carlin, you could come up here and give the talk reciting your own history, but up until 1983, there was actually no option for these babies, and they typically would not survive more than a few days. And then in 1983, Bill Norwood, whose name is now the name of the heart surgery that's done to palliate these children, published this paper that actually shows what he does actually over 3 surgical steps. And effectively what you do is you convert the right ventricle to be hooked up to the aorta, so that becomes the main pumping chamber of the heart, taking over the function of what was supposed to be the left ventricle. And then the other 2 stages show here that the blood that was supposed to normally return to the right ventricle to get blood pumped to the lungs, though that blood actually gets reconnected directly to the lung arteries. So that's called the Glenn procedure and the Fontan procedure, right, Carla? Yep. And it's so much fun to be able to interact with my audience. I never could otherwise. And so basically what you're left with is a 2-chamber heart. And in fact, whether it's a hypoplastic left ventricle or a hypoplastic right ventricle, whether it's aortic and mitral atresia or just stenosis, this is all kind of the common end pathway. So this is why, from the clinical standpoint, everyone kind of lumps this together. But again, when you think about the actual specific disease process, there's a lot of variability between patients. So how did I get started with this? So I think I mentioned the other day that my first month of cardiology training connected me with a patient who had this chromosome anomaly that most of you probably have not even seen what this is. This is the old-fashioned karyotype. And I think it's clear that you can appreciate that compared to the normal chromosome, there's a chunk of this chromosome missing. So this was back in 1995, and it didn't take a rocket scientist to figure out that if this is the cause of the hypoplastic left heart syndrome in some of these kids, then there must be a gene or genes in this region that's missing that's responsible. And so that's really what I've been working on now for the last 20-something years. And that allowed us to begin our studies to learn much more about the syndrome. And so we published this in 2004. Prior to that time, the largest series was 17 patients. So we published a prospective study of 110 patients, which is still being referenced this day as a source of information. And in terms of the heart defects, here's kind of a summary of what we learned. So if you take all comers with Jacobsen syndrome, about half have some kind of a major heart defect that usually requires surgery. And what I learned is that there's actually probably an unprecedented high frequency of hypoplastic left heart syndrome that occurs in Jacobsen syndrome, maybe as high as 10% of all babies that are born with hypoplastic left heart syndrome, which in reality probably accounts for somewhere around 1 to 2% of the 1 to 2,000 babies born every year in the United States with hypoplastic left heart syndrome. And of note, what's really important from a research is in most cases, but not all, full-blown hypoplastic left heart syndrome in Jacobsen syndrome is of this anatomic subtype where the two valves are completely blocked off. And I can tell you, if you look at the literature, almost nobody has focused on trying to dissect out the mechanism of a specific anatomic subtype. They just kind of lump everything together. But I think, as I showed you before, if you try to lump together two very different mechanisms, you're probably going to create more confusion. So many of the major heart defects that occur in Jacobsen syndrome also occur in the general population, and that's really important because we've already begun to see parallels between other patients that don't have Jacobsen syndrome, but may have this common underlying mechanism that we're beginning to define. And I'll go over a little bit to begin to answer why is it that you can have two people with the same deletion, one with hypoplastic left heart syndrome, and one with maybe a hole in the heart, and another with a completely normal heart. So I think I showed you the same slide the other day. This is a technology that most of you are probably familiar with, what we call microarray or array or CGH, comparative genomic hybridization. Anyway, it allows us to really pinpoint at the molecular level where the deletion is, and that is actually really helpful because we've been able to take advantage of different deletion sizes that allow us to kind of pinpoint where we think the gene is, and this is just a cartoon kind of showing that. So this blue here, this black represents just chromosome 11, the end of the chromosome there, and so this blue represents the smallest deletion that we know of a child with Jacobsen syndrome and hypoplastic left heart syndrome. Even so, that blue area contains maybe something like 50 genes, so that's still pretty challenging to try to figure out which of those 50 genes. But what really helped us is even rarer than this is the occasional patient who has, instead of a deletion going to the end, a deletion within the chromosome. So we call that an interstitial deletion. And I think you can appreciate here, if we assume that it's the same genetic cause in this patient as in this patient, then we would hypothesize that it's this area of overlap, or even smaller here, that contains the disease-causing genes. And that's exactly what we've been able to do. And thanks to the Human Genome Sequencing Project, which came out in the early 2000s, we could take advantage of that to determine that there are about 6 genes in that minimal region of overlap, 2 of which we think are very, very important. And in fact, one already had been shown in animal models to have a critical role in heart development. And more recently, we've identified a second gene that actually is related to this first gene that we think may actually be the cause of hypoplastic left heart syndrome and Jacobsen syndrome. So these 2 genes are what is called a transcription factor. The transcription factor is a gene that basically regulates the expression of other genes. So it turns on and turns off genes at critical times, either in development, like in the heart, which I'll tell you about, but also when these genes are abnormally expressed in the wrong place at the wrong time, let's say in one of us as an adult, a lot of these genes have also been shown to be implicated in either causing or exacerbating certain forms of cancer. So it begins to give you an idea of how complex this story is as we begin to unravel things. So most of what I'm going to talk to you about today is our studies on this gene. But again, we have some recent evidence that we think also implicates this second gene, slide 1. And so we published this paper that actually showed a patient with a different type of heart defect in which the left ventricle was small, who did not have a deletion in chromosome 11, but actually just had what we call a point mutation, so just one letter of the genetic code that was altered in the ETS1 gene. And so that provided further evidence that ETS1 must have a critical role in causing some of these heart defects, we think, including affecting the development of the ventricle. And then, as I alluded to, much more recently, we actually have a patient here in San Diego who has hypoplastic left heart syndrome, who has the same anatomic subtype with atresia of the aortic and mitral valves, and also has the bleeding disorder, Paris-Trousseau. And this patient does not have a deletion, but this patient has a point mutation, in that same gene that I just mentioned, FLY1. So this actually adds to the complexity, but may help us to explain the specific cause of hypoplastic left heart syndrome, and I'll come back to that in a moment. So in my lab, we use genetically engineered mice to study these genes that are involved in heart development. One, because mice have most of the same genes that we have. So we're about, 90% identical to mice, so we only differ by 10%. The other thing is that the mouse heart is very, very similar to a human heart, and so it's a really good experimental model system. And whenever you're studying any gene that you think is important for whatever function, in this case, heart development, you want to determine where and when this gene is functioning to give you some clues about what it actually might be doing. And in this case, it turns out that the heart is not just one type of cell. There are a lot of different types of cells that have all kinds of different functions, whether it's the muscle itself or the valves or the inner lining, the outer lining, the vessels, et cetera, et cetera. And what we just are showing in this picture, don't worry about the details, it's just to identify, shown in the blue, that there are actually two populations of cells that ETS1 and actually FLY1 too are expressed in. Interestingly, they're not expressed in the heart muscle cells, so the cardiac myocytes, the actual functioning cell. They're actually expressed in two different populations that we also know are critical, which I'll talk about in a moment. One is the neural crest, which I think I mentioned briefly the other day, and I'll go over that more in a moment. And the other one is in what is called the cardiac endothelium. And there's actually two populations of cells that we refer to as the cardiac endothelium. One is shown here, just the inner lining of the ventricle, and that's called the endocardium. And the other one that's not obvious here is actually the coronary arteries are made up of very specific types of cells called endothelial cells. And that's also where ETS1 is expressed. And I'll come back to why that is so important in just a few minutes. So this is kind of summarizes what I told you, and that is we hypothesized then that depending upon which heart defect we're talking about, it's the loss of ETS1 and maybe FLY1 as well, in one or both of these populations of cells that may be responsible for these different heart defects. And this is just kind of a cool classic slide, just summarizing what we mean when we talk about neural crest cells. In this case, we're talking about cardiac neural crest cells. So this is just a cartoon showing the developing spinal cord, basically, in a mouse or a human. And it turns out there is a subset of cells called the cardiac neural crest cells that, as the heart and the embryo are developing, these cells actually migrate out from the neural tube or the developing spinal cord, and they, in a very precisely regulated way, migrate out to go to help form some of the final structures of the heart, including the outflow tracts. So that's what separates the ventricles. So what's it called if you have a deficiency of that wall between the two ventricles? Hole in the heart or ventricular septal defect. Other cells are critical for the development of the valves that are in the aorta and the pulmonary artery, so I'll come back and talk about that, and other cells are important for other parts of the aorta. So this is actually an exquisitely complex population of cells that, as you can imagine, have to perform perfectly to help form those permanent structures of the heart. 99% of the time in the general population, it works perfectly, but for the 1%, I'll tell you more about how we think those are causing some of the heart defects in your children. Interestingly, in hypoplastic left heart syndrome, just some circumstantial evidence further implicating neural crest cells. There's actually a problem with the neurons that are responsible for regulating movement of the GI tract, of the bowels. That disease is called Hirschsprung's disease, and that's thought to be a neural crest cell defect. And interestingly, in hypoplastic left heart syndrome, all comers, there's a much, much higher frequency of this type of neural crest disease called Hirschsprung's of the GI tract. And so that kind of strengthens the case that neural crest cells may have some component that contribute to the pathogenesis of hypoplastic left heart syndrome. This is a picture of a mouse embryonic heart that we have genetically engineered in my lab. This is actually a three-dimensional cardiac MRI showing here a normal mouse heart. This is 1 millimeter, just to put it into perspective. And here you can see the left ventricle with the aorta connected, so blood goes out there. Right ventricle, that right there is the wall that separates the two ventricles, so that's the ventricular septum. And if you look at a pathological slice through that, these are the three leaflets making up the aortic valve, and those blue dots are actually the neural crest cells that are part of the development of that valve. This is kind of another way of looking at the same thing. You can see here the red dots. Those are, again, the neural crest cells. And when we eliminate through some genetic engineering tricks, the ETS1 gene, just specifically in neural crest cells, what we get now is one of the heart defects that you see in your children, not hypoplastic left heart syndrome. What we see here is a hole in the heart. And rather than the aorta being lined up with the left ventricle, it's actually lined up with the right ventricle. So when you have a hole in the heart and the aorta lined up in the right with the right ventricle, we call that double outlet right ventricle. And in about half of the mice that we studied, rather than having the normal 3 aortic valve leaflets, they have only 2. And compared to that, you can see almost a complete absence of that blue, as shown here with a significant decrease in those red dots, suggesting that part of the reason for this is due to, for whatever reason, an absence of neural crest cells in this finalized aortic valve structure. And we can actually look at this up close. So as I showed you in the cartoon, those cells have to migrate to form those final structures. And this is just a normal heart. And you can see this kind of streaming path of neural crest cells migrating from the neural tube towards the heart. And when we eliminate the S1 gene in our mice, specifically in neural crest, you can see how we just kind of lose that process. And so we think the mechanism is probably due to a defect in the cell's abilities to migrate to get out to that developing heart. And in fact, this is what we're talking about, is this population of neural crest cells that go all the way down here to form part of that wall that separates the two ventricles. And so one of the things we were able to do is actually derive neural crest cells from our patients with hypoplastic left heart syndrome. Now remember, your child has a deletion of 1 copy, but also a normal copy. And so what we wanted to look at is in the patients that have hypoplastic left heart syndrome with Jacobsen syndrome, we can, through some trickery that I'll show you in a minute, recreate neural crest cells and study them like in a test tube. And one patient, it turns out that compared to controls, that one supposedly normal copy of S1 is not being expressed at all. So that basically indicates that there's a complete loss of S1 expression, which is kind of like our mouse knockout models. And that might explain why, in that case, with a complete loss, among other things, you get some type of a heart defect. In the other patient that we looked at, there is a normal expression level, but we think the function of that one version of the S1 gene is abnormal. So 3 different possibilities: either complete loss of S1 from the normal chromosome, or a copy of S1 from the chromosome but doesn't function normally. That might explain why you get other types of heart defects, and I'll come back to that in a moment. Or lastly, which we don't show you, well, here is normal versus probably in your children that don't have a heart defect, you would see something like these for that one, but in this case, expression, normal level of a normal gene, a normal protein. All right, so that's the neural crest cell story. Now I want to tell you what our lab is really focusing on as well, and that is, as I mentioned to you before, what is S1 doing and Fly1 doing in this other population of cells. And this, I don't know if you can see this little green line, this just kind of shows you where this inner lining of cells that S1 is expressed in, called the endocardium. And it turns out, I'll come back to that in a moment, the other area is this is showing a little tiny branch of a coronary artery that is made up also of these endothelial cells. And what previous studies have already shown is that this population of cells, either forming the inner lining of the ventricle or the vessels, the coronary endothelium, actually secrete factors that are critical for the development of the ventricles. And so that is beginning to guide our future studies, trying to determine what are these signaling pathways that S1 and Phi1 are involved with that are so critical for regulating the development of the ventricle. And so to do that, we take advantage of several different animal model systems. This one, actually, believe it or not, is a heart of a fruit fly. Yes, that little thing that flies around in your kitchen and is a nuisance. And in this case, this is an embryonic fruit fly heart. I don't know how well you can see that, but it's just like a little tube of cells. And even kind of like a mammalian heart, there are different cell types. One type is the yellow cells here, and the other is the green. And even before I came on board, this paper was discovered in 2003 that showed that in the fruit fly, the gene, that's the fruit fly version of S1, called pointed, when that's mutated, look at what you see. In this case, in abundance, too many of these heart cells that effectively kind of clog up that pumping chamber. So this is like the normal pumping chamber, essentially like the ventricle. And in this case, it's completely clogged up. So that was really interesting, but you could say this is hypoplastic left heart syndrome in a fruit fly. What does that have to do with the human disease? Well, another model system that's a little more like a human heart is a frog heart. And in this case, this is a normal frog heart. So this is these green, everything that's seen here in green is just showing normal heart cells. So this is a normal frog ventricle. And if we specifically eliminate the S1 gene in basically the cells that become the endothelium, you get this kind of a ventricle. So that is very dramatically different from that. So it's a small ventricle, and if I had a video, I could show you that compared to this nicely contracting ventricle, this thing barely moves. So essentially what we've shown here is that eliminating the S1 gene in the cells that become the endothelial cells in a frog heart cause a small ventricle that doesn't pump well, and that's exactly, those are really the hallmarks, as I told you, of hypoplastic left heart syndrome. What's been frustrating is that when we set out to do the same experiment in the mouse, we have never seen hypoplastic left heart. So if we try to take out just S1 in the cardiac endothelium, we do get something that's very abnormal. So in this case, this is just taking a look at that left ventricular wall. So there are actually two populations, this kind of compact zone, and then this more finger-like layer called the trabecular layer. And what we see in the knockout mice is not hypoplastic left heart, but something else in which there's almost a complete loss of this compact zone, and then you just get these very lacy looking, very skinny finger-like projections. And in this case, this was so abnormal that the mouse couldn't even make it to delivery. So this embryo died in utero. So it's not hypoplastic left heart syndrome, but it is some kind of a defect that we actually call non-compaction of the left ventricle. So one of the questions that we're trying to understand now is, why is it if we knock it out in the frog, or even in the fruit fly, we get something that looks like a hypoplastic left heart, whereas in the mouse, when we knock it out, we get a different type of defect in ventricular development, as shown here, called non-compaction. So that's a lot of what we're focusing on now. But having said that, one of the things that we can do is to take advantage of the molecular tools that we have to try to define, as I mentioned to you before, these transcription factors turn on and turn off specific genes at specific times during development that are critical. And what we can do is actually use some very powerful molecular tools to identify what those so-called downstream factors are that S1 are regulating. And so Lou in my lab has identified a whole list of these genes. And what's really, really interesting is these are the genes that S1 directly bind to, like a molecular switch to turn on, or turn off these specific downstream target genes. So for example, this gene called ALK1 has been shown already when there are mutations in ALK1 in human patients to cause total annulus pulmonary vein. In other cases, in a frog heart, one of these other downstream target genes called claudin-5, when that's knocked down, you get a hypoplastic ventricle, just like what I showed you. And then in other cases, like where's Jamie is in the room, this downstream target gene, when has been shown many times when it's mutated to be associated with tetralogy of below. So what we think is actually going on here is for any one patient, each individual patient may have much more sensitivity, let's say, to the loss of S1 on the expression of this gene to give you anomalous veins versus, let's say, this gene in a different patient, let's say in Jamie, to give you tetralogy of below. So they think that's beginning to help us to understand why we see these different heart defects in different children. We haven't proven it, but that's what we think. And so just to kind of summarize, this is the young man that I first saw in 1995 that I still take care of. So he is my most historical Jacobsen Syndrome patient. This is the little girl from Colorado who died about 15 years ago who had that much smaller deletion that helped us to narrow it down to 50 genes rather than over 100 genes. This little baby had a very, very tiny deletion that implicated S1. She was born with hypoplastic left heart syndrome and passed away at about 6 months of age. And I show you these three precious babies that we have come to know: Sophia, Thor, and Sebastian, who all passed away in the last year, whose hearts now we are studying, because ultimately we can come up with all these great hypotheses from our animal models, but we have to close the loop to determine if that's indeed what's going on with the human hearts. I'm just going to very briefly say, and I'm going to come back to this story in 2 years, but we've actually found, switching gears here, another gene that potentially, when mutated, can cause a lot of the same problems that occur in Jacobsen syndrome, including, in this case, a patient with hypoplastic left heart syndrome. This gene is not on chromosome 11. So we haven't proven it's the cause, and those studies are underway now, but it may be that this gene has some critical role in regulating the expression of the genes in chromosome 11 that we've identified. So stay tuned. Okay, I'm going to show you a little movie. I hope the volume will come on. Curtis, if you can help me with that.
If not, you can put the microphone over there.
Let's see. I may have to. I need help. Someone help me. Why is there no volume?
Where's Katie?
I can go back. Let's see. Just have to coordinate our efforts here. Okay, let's see what happens. Well, anyway, that's okay. I can sort of narrate this, but what this is showing you is just the technology that I alluded to you the other day called iPS cells, or induced pluripotent stem cells. And what this is I could take a piece of my skin, or actually these days blood cells, and actually through some recently developed genetic trickery, reprogram those cells to become stem cells or pluripotent stem cells. And then through some further genetic trickeries, we can actually reprogram what were originally skin or blood cells to become almost any cell that you're interested in. So what cell types, what might we be interested in our case? Obviously, heart cells, like the cardiac myocytes or endocardial cells. And so we are using this technology to give us clues about what goes wrong in the human patients, studying human cells that have a specific genetic cause, like Jacobsen syndrome, or mutations in specific genes in chromosome 11, that we can then study to see maybe what's wrong. I already showed you one example of how we use that technology looking at neural crest cells. And so how do we use how can this be helpful? Well, I'm going to show you in a moment an example, because I think right there that demonstrates profoundly the difference in outcomes in children with hypoplastic left heart syndrome. And to be honest, for the most part, when a baby is born with hypoplastic left heart syndrome, we're not really smart enough to predict who's going to do well versus who is going to have problems early on. And if we could determine that from the get-go, then we can do a much better, more sophisticated way of sort of personalizing the care. And so using this technology, we can create cardiac myocytes that will allow us to perform these sort of functional studies. Just to kind of summarize, it will allow us to sort of stratify or identify those conditions that may develop early heart failure. In Sebastian's case, I think what we did not appreciate as much as we should have is that even though his left ventricle looked like early on it was actually functioning pretty well, the fact was it was not able to grow very well, so we got a little bit fooled. And if there's one thing we learned from Sebastian and others, is if we could do some kind of a study early on to be able to predict the growth potential of that ventricle, we probably would have done something differently. And the group from Mayo Clinics that some of you may have heard of is actually using stem cells that they think might be able to help to promote good function of the right ventricle. But not all patients respond the same way. Some are better candidates than others, and we can use our technology now to identify such patients. And ultimately, we might even be able to use this sort of technology to create or discover new drugs that are specifically targeted for right heart failure, of which we have almost nothing available today. And if you look carefully, this is what I'm talking about. So this is actually an artificial pumping chamber that is derived from human cardiac myocytes from a human patient. So I think you can appreciate here that this little windsock-like thing is actually doing what a left ventricle is supposed to do. And with a lot of really sophisticated technologies, we can put this pumping chamber through a whole battery of tests, again, from a newborn baby, that will give us potentially a much better understanding to predict how that baby is going to do and what kind of personalized therapies are going to need. So it's not a heart, so there are definitely limitations, and the biggest limitation is, well, the advantage is the great way to assess function and some of those other things I just mentioned to you, but it doesn't give us really, an opportunity to learn about what goes wrong in development, right? So I think one thing you can appreciate from what I told you is that we still don't have a great model for recreating hypoplastic left heart syndrome in an animal. But having said that, I want to tell you about a very, very exciting new technology that's just beginning to come into its own. And I don't know how well you can see this. I think like yeah, so you may have to turn down the lights a little more, but if you look really carefully, what I'm showing you here is an attempt to recreate a heart in a petri dish. Now, this is not a heart, but what this is a collection of cells derived from a human patient, and through, again, some genetic trickery, they are programmed to become heart cells that actually can begin to function like a heart. And I don't know if this comes out too well, but let me just see if I can get it to go. But basically, unlike just what I showed you before, this we think is representing a very, very early stage of heart development that we can then take advantage of if we derive the same type of what we call a cardiac organoid from specific patients. And how we're going to use that is as follows. So we already know a couple of genes, actually 3 genes. I didn't talk to you about the third one on that list. So we have a patient with an S1 point mutation, which is a patient with a FLY1 point mutation. And we have another patient with what we think is a different downstream target that are all involved very early in heart development to cause hypoplastic left heart syndrome. And the idea is we can, just like for a normal control, we can generate a cardiac organoid from, let's say, a patient with hypoplastic left heart syndrome and mutation in any of these genes, or like in Carlin's case, the deletion. And what we think is, compared to what I just showed you, that blob of cells, if we're right, we might predict then that when it's derived from a patient, maybe that blob of cells will not develop or function normally, so it might be smaller and not contracting as well. And then ultimately what we can do is to prove our hypothesis that, for example, if we think it's FLY1 that's the cause, We can then go back and use some further genetic trickery to just correct that fly1 mutation in that patient, with the idea that once we do that, we then generate another cardiac organoid and show that now it's functioning like a normal one. And if so, that would give us something that we've been so desperate to go after, and that is to have a really good experimental model system to study hypoplastic left heart syndrome from some of the earliest stages of heart development. Okay, how am I doing for time here? Good, we're just about at the end. So as a different, just to change gears a little bit, just to tell you how we can sometimes take advantage of other rare patients in our practice. This is an ultrasound of a fetus that was found to have a hugely dilated left ventricle compared to the right ventricle. And in this fetus, the baby was found to have a complete absence of the aortic valve. So in that case, if you can imagine, every time that ventricle pumps blood forward, what do you think happens if there's no valve ability to close? It just leaks right back. And you can imagine that's not a good situation. That's not compatible with life. And this is why this left ventricle was so humongously dilated, basically taking up the whole chest of the baby. That baby did not survive. But what we were able to do is get some genetic material to do whole genome sequencing. And we were able to actually identify what we think is a disease-causing mutation in a gene called APC. Don't worry about what it stands for. But it turns out that in cells, this APC gene is absolutely essential for cells to survive, okay? And it turns out even more specifically, remember I told you that neural crest cells are critical for the development of the aortic valve? Well, it turns out that this APC gene is critical for maintaining neural crest cells. So we think that the loss of this gene led to a specific loss of cardiac neural crest cells that are required for the development of that aortic valve. And so Marjan in my lab, who joined me about a year ago, is working on trying to figure out exactly what this gene is doing and in what specific subset of cardiac neural crest cells that are required, in this case, just specifically for the development of the aortic valve. And remember I told you before, for S1, it's this loss of neural crest cells that are making up the wall that separates the ventricles. In this case, we think it's the loss of neural crest cells that are just specifically required for making up that aortic valve. All right, so just to kind of close, I wanted to come back to a few of these questions that I mentioned to you before. I think it's complicated, but I think we're beginning to now gain some insights into why some of your children are born with a heart defect, some not, and why some may be born with hypoplastic left heart syndrome versus total anomalous veins versus tetralogy of Fallot. And lastly, again, I think every so often on the Facebook chat or whatever, we see people asking, or is it really true that there's a much higher frequency of red hair in kids with Jacobsen syndrome? What do you think, Teresa? Do you notice that in Italy?
No.
Yeah, well, you've got darker hair anyway, thank you. In the United States, we have a more of a blend, so diversity is great. Pardon me?
More people. I'm sorry.
Right, so we have that, but what I can show you is that when we eliminate S1 in a different population of neural crest cells, compared to the normal mouse, you can see here these mice have big white patches of fur, and it turns out X1 or neural crest cells are also involved in the pigment formation. So in skin cells that are required for pigment called melanocytes. All right, this mouse doesn't have red hair, but it has white hair. So I think it suggests that maybe it's the role of S1 in melanocyte function that might be one contributing factor, as well as Irish genes, perhaps. So it's not the only one. All right, I want to finish by acknowledging again the people that are just so critical in my lab. So if I can just take a couple minutes. So this is Tai Chi, who actually is a friend and classmate of my son's dating back to kindergarten and teammate. He is going to be a senior in high school. And Virgil, if you're here, you'll be fascinated to know that his grandfather in Japan is the one who discovered T suppressor cells. I Googled it, I'm not quite sure if I was able to find it, but Okamura is his name. And actually, Tai Chi is hoping to pursue a career as a physician scientist. He's going to be a future pediatric allergy immunologist, hopefully. He is actually working on a really cool project with a lady by the name of So Young, excuse me, who is now just finishing her PhD in molecular biology at UCSD. She wants to go on to go to medical school and pursue a career as a physician scientist. Enrique Rodriguez is a really super smart, super nice young man who is going to be a senior at UCSD studying biology who wants to go to medical school. So he's just starting in my lab. This young lady, Annie Ting, has just graduated from high school and has been accepted and will be going to Duke University to pursue a path in medicine. You met Lu Wang and Marjan Hassari, who are Lu is a PhD, Marjan is an MD, who are doing the bulk of the work that I just showed you. In addition to the people in my lab, I'm really incredibly fortunate to work with other principal investigators, PhDs. So this is Karen Okor and her husband, Ralph Bodmer. They work on the fruit fly and zebrafish. And I can tell you their work led to the discovery of a gene called tinman in the fruit fly that in the fruit fly, if you mutate that gene, they don't have a heart. But they are amazing because they are so genuinely concerned about how their research could impact clinical care. And it turns out there's the human version of this gene called NKX2.5. And because of their work single-handedly, we have identified patients with mutations in that gene that has led to a life-saving intervention to put in a pacemaker. And without a pacemaker, some of the families that I take care of now would not be alive. And that all started with their incredible work on fruit fly heart development. There's Lou again. And then Shui Nai is an incredibly talented PhD biologist at Georgia Tech, whose work I showed you on frog heart development. And so I think what's really, really gratifying to me is we have this incredible multidisciplinary group of people who, first of all, we all love each other and get along great. But what is, I think at the end of the day, so important is we all recognize that what we do together would be impossible for any of us to do individually. This is the first baby I diagnosed with hypoplastic left heart syndrome. She does not have Jacobsen syndrome, so she is going to be 28. I think I diagnosed her in July of 1996. So, the way I kind of look at it, the people I just showed you from my lab, they're kind of like the engine. I'm kind of like the steering wheel to make sure it goes forward and in the right direction. And actually, you guys have been sort of the fuel, the gas that allows us to keep going through your incredible fundraising efforts. And so, again, I want to thank you because everything I just showed you would be almost impossible without your support. And I will forewarn you that every year, and again this year, I will reach out to you for more fundraising support and efforts. Even simple things like reaching out to your friends and family. I can tell you I'm really shy. I have never felt comfortable begging for support for my research. But I will tell you this, and I think Craig and Rebecca could really attest to this, and many of you in this room, that when you reach out to your friends and when you explain to them the personal interest that you have in this, you will be amazed by their response. And people will rally and come to support in ways you never could have imagined. And it's just like what Ryan said the other day, whether it's for fundraising or dealing with the other issues that you have to go through with having a child with a disability, you just have to ask. And when you make that ask, you will be absolutely overwhelmed by how people can step up and rise to the occasion. So thank you again. No one's asked any questions yet. Okay, good. All right. Claire, go ahead. I can recite your question if you like.
At the end of the year, when your link comes up.
Yeah.
For, end of the year.
Fundraising campaign, yeah.
It's sometimes unclear. I think it looks like it goes directly to rating. And I've had to ask you a couple of times.
Right, so sometimes it gets a little confusing. The question is like, what's the easiest way to make a donation? So just to explain to you, each year starting in November, so basically November into January, we have what's called the Light the Way campaign. So it's a specific campaign just for the end of the year. And the Marines Foundation, we met Elisa the other day. They're the ones that oversee that. So it's supposed to be as simple as a one click and you get right to the page that you populate to make your donation. So we do run into some bumps, but that's how it's supposed to be. It is designated not to go to Rady Children's Hospital. It is designated to go specifically to my research lab. And what actually happens is that money gets kind of saved up in an account at Rady's, and then once the season is over, it all gets transferred directly to UCSD to go directly to my lab. And the other thing that's great is there's zero overhead. So every dollar of your donation goes directly to support my research. Teresa.
I want to give you some suggestions. Let's start from the beginning. It's clear that Fly1 is responsible for hypoplastic left heart syndrome. This is okay.
Well, we hypothesize, but
Yeah, this is
it's
okay.
pointing towards that.
It cannot be the only thing, because otherwise all Jacobsen would have the same condition. So there must be something else. And you mentioned the possibility of the non-deleted chromosome to be responsible. That's one. I think you should think also of the Alpha syndrome is associated also with left heart syndrome. So maybe there is a combination of genes outside chromosome 11, and you can have already an idea which ones, homobox genes, because EPS1 and FLI1 are transcriptor factors. So there may be the combination of, so the best to do is first check FLI1 in the normal chromosome 11. Second, NGS of all the genes connected with heart.
So the way we're addressing that is our hypothesis is in the frog, if you just knock down ETS1 or Claudin-5, you get a hypoplastic ventricle. I didn't have time to, what I gave you is a fairly superficial overview. I could spend an hour. So what we think in the mouse and the human is you actually have to have ETS1 and FLI1 both mutated or deleted. What we know is, and I didn't have time to show this, but I have a beautiful schematic diagram that shows that ETS1 regulates FLI1 expression and FLI1 regulates ETS1 expression. And we're actually making, as we speak, a double mutant in the mouse of just ETS1 and FLI1 that we're going to delete just on the cardiac endothelium using FLOX alleles and CRE drivers that are specifically just for the cardiac endothelium. And our hypothesis is that if you make a global knockout of FLI1, they die very early because of bleeding in the brain. But if we can just restrict its loss to the developing heart, we hypothesize, although there could be other factors that I'll come back to in a moment, we hypothesize that might give us the hypoplastic left heart phenotype in the mouse. Having said that, to your point, I already showed you that we know that there are very numerous downstream factors that are part of this very complex, what we call a gene regulatory network. A gene like TBX5, which is the gene that causes whole organ, which has been on rare occasions associated with hypoplastic left heart syndrome, could be one of those genes. And so, at the end of the day, what we think is that FLI1 may be regulating additional genes besides what ETS1 is regulating. Maybe TBX5 is one of those, and we'll find that out as we do what we call single-cell RNA sequencing to actually analyze the gene expression pattern of all 23,000 genes from a specific cell, an individual cell, one cell from a specific point in time. And so, you know, that's a bit of a long-winded answer, but yes, we are all over that, I guarantee you. It just was not quite amenable to going through that much detail in 1 hour or so. So, anyone else have any questions? Yes, right, yes. So the question is, Jamie has a variant of what I showed you, double outlet right ventricle. This gets into some real nuance here, but basically the variant is similar to double outlet right ventricle. So the two kind of go together. So there is what we call a tetralogy-like version of double outlet right ventricle, and that's actually what we see in our knockout mice. So our knockout mice pretty much have exactly what you were born with, Jamie. How cool is that? Does that answer the question? Great. Any other questions? No? All right. Well, again, I just want to thank all of you, and a special thanks to the Servant family for being here. Carlin, thank you for letting me use you as a profound, wonderful example of why we're all here and why I do what I do. And if I said anything wrong, please let me know, because you are the expert, okay? All right. Any other questions? All right. Thanks.